So today I'm going to talk about comparative analysis of target RNA-seq and optical genome mapping for detecting pathogenic rearrangements. So the biggest issue in our line of work as a laboratorian is conflicting results. They are problematic. And unfortunately, we are seeing more and more with using multiple testing modalities. So in this study, we were comparing OGM and RNA-seq in acute leukemia. So the formal question that we were asking is, what's the concordance rate between RNA-seq and OGM for detecting gene rearrangements and fusions? And is this concordance rate is changing to type of leukemia, the gene, or the underlying genetic mechanism? So our study group consists of 467 acute leukemia. And the sample mix is AML, BALL, TALL, and MPL. It reflects what's being seen in our cancer center. And I think based on the prevalence of these diseases, this cohort is representative for all acute leukemias in terms of the frequency. And the test that we were comparing is optical genome mapping versus leukemia translocation panel. So leukemia translocation panel is a 100 gene in-house developed panel. And this is basically Archer chemistry. So the underlying basis for this test is if one of these 108 genes are involved, then the chimeric fusion transcripts would be detected. That's the idea. So all new findings are not equal. And for that, the TR1 findings are the findings which has diagnostic, prognostic, or therapeutic importance. TR2 findings have some clinical relevance, but they are not meeting TR1. And TR3 findings they are acquired, but its clinical significance unknown. So our overall concordance rate was 88%. But it shows differences across malignancies. For example, the concordance rate for AML was 91%, whereas for TALL it was 53%. What you are looking in this figure is that the green ones are negative by both OGM and Archer RNA sequencing. The purple ones are the concordant findings, both with OGM and Archer. Red is only OGM only, and aqua is only Archer. So this is basic data table, which shows the distribution of detected genes aberrations according to the TRN methodology. If you look at, there is concordance for most of the cases, but if you look at genes like MECOM, only 26% concordance, or BCA11-1 has only 0% concordance. Concordance means in that setting is that positive by both methods. And this very same thing is happening for IGH findings too. So what are the underlying genetic mechanisms associated with discordance? So one is enhancer hijacking. The second one was insertions. And the third one was intrachromosomal recombination. So when we look at the genes that is involved in enhancer hijacking, the concordance rate differs significantly. Only 20% of the genes is concordant between OGM and Archer-based next-generation RNA-based sequencing. And these are always the OGM positive cases. So genes like MECOM for BALL, IGH for BALL, and BCL11B for TALL were the genes that is clinically significant for those diseases, but they are blind to RNA-based sequencing because RNA-based sequencing only focused on chimeric fusion transcripts by its design, whereas with OGM, it is possible to appreciate the underlying genetic aberration. So this is a case for enhancer hijacking. So what we have here is 514 translocation. And as you see, the Archer assay, no fusions were detected. When we look at this case, we see the involvement of the TLX3 on 5 and BCA11B on 14. Obviously, if you're in the know, someone can say that, I mean, BCA11B is not aligned. I mean, it is the other side of the fusion junction. How can we say that BCA11B is involved? And that would be a very good question. The reason BCA11B itself was not involved, but BCA11B enhancer was involved. And that BCA11B enhancer was driving TLX3, presumably TLX3 overexpression. And since it is enhancer driven, there were no fusion transcripts detected at RNA level. There were no chimeric fusion transcripts detected at RNA level. Another thing is insertions. They are bane of cytogenesis that's very difficult to ascertain. In this case, we got fish finding. We have one green, one fusion pattern, which may be compatible with a KMT2A rearrangement. And we have a definite KMT2A rearrangement by the RNA-based fusion assay. But the initial look at OGM data confounded us. But when we look at data in detail, we are seeing some yellow stripes which were not aligned. So we looked at the underlying reason for that. And for this gene, KMT2A AFDN fusion was the underlying aberration. So when we manually align those yellow stripes, which were not mapped to any region, it was perfectly aligning manually with the pattern in the AFDN gene, which you can appreciate by the red rectangles in upper and lower panel. And also, if you look at these findings in more detail, it was exon 8 and exon 2 fusion, KMT2 exon 8 and AFDN2, which is apparent also from the OGM studies. Moving on. Now, another insertion concerning with KMT2A, but currently the discrepancy is not between OGM and other techniques, but fish and fish cytogenetic assays and the molecular assays. Because neither fish nor chromosome analysis indicate the KMT2A MLL10 fusion detected by the molecular assay. And if you look at the fusion, we have is intact fusion signal. This is not consistent of a rearrangement of whatsoever. But what is remarkable is that thing is very hard to ascertain here, but was on chromosome 10. Actually, for this case, OGM was very helpful. So what we have here is, this is the green bar is the KMT2A, the blue bar is the is the consensus map, and the red is the reference. So what we have is that there is KMT2A is here, KMT2A is here, and in between it is MLL10. So it is an unequivocal demonstration of the insertion by optical genome mapping. Now, intrachromosomal recombination. I mean, intrachromosomal recombination are fusions due to intrachromosomal recombination is happening probably more than we think. So I'll be talking about two cases with the P2YRA CLF2 discordant results. So this case had flow cytometry CLF2 was negative. And the molecular assay is showing the fusion, which is in frame, but it's just 6-7 rates, not that much. And based on the OGM data, there is no way to ascertain the existence of that fusion. On top of it, we got a known driver for a BLL, ETV6 RUNX1 rearrangement, which is both detected by optical genome mapping and RNA fusion assay. Most likely, there is a very small clone detected by the molecular assay, but the flow was negative. It's below OGM, so basically, honestly, not probably not very important in the pathogenesis of the disease. A very small subclonal aberration. Now, a second one. Now, this is different. So now we got about more than 100,000 reads, and the fish is consistent of rearrangement. I mean, it's not a typical signal pattern, but what we are seeing is one yellow, two green signals, perhaps some sort of an unbalanced rearrangement or a deletion. Now, the OGM data is interesting. So let's look at much more detail. And this is the later case. So I have learned my lesson for the AFDN one, right? AFDN KMT2A. So when we are looking at this stuff, these band patterns are highly similar to this one. And so I can see those blue stripes. Perhaps this one is that one, but there is no way that, honestly, neither the computer or I'll make the call unequivocally based on this information. But probably the underlying data is there, but it simply is not detected. And for this case, we also have the CLF2 overexpression by flow cytometer studies. And this is the last case for intrachromosomal recombination showing a NUP214-ABL1 fusion. This is a very interesting case because NUP214-ABL1, there's only 37 reads. It's not that much. And when I look at the OGM board for the SV annotation, it's not there. But it's okay. Just for the heck of it, I actually make all the settings less stringent. Obviously, it may give you false positive results, but it is also giving you something. And able to see the NUP214-ABL1 rearrangement. I mean, based on what is being happening here is probably this part is during the recombination is inverted and comes here, and this is consistent with that data. But I was curious why we are not seeing that. And one of the strengths of the OGM is also the supporting molecular data. I mean, here's the supporting molecular data for this consensus map. And when you look at this, I mean, it is evident why we don't see this blue consensus map. Yeah, they are there, but there aren't many molecules that bridge, from NUP214 to ABL. I mean, I see this one perhaps, and perhaps this one. Only the two molecules. I mean, there is no way that it should be, it can be called. But as an exercise, it also shows how much is under the hood. And when we are looking to the data, we should be really careful and taking advantage of the technology stuff. So, what's the limitation of these studies? Well, this is a single institution study in a major academic center. Well, MD Anderson is a very big cancer center, but other experience in the world may be different. I mean, this is just how things are seen in our neck of woods. Also, we compared only, this comparison was only done for 108 genes. I mean, I wasn't talking about the copy number gains or losses or aberrations, not involving these 108 genes for apples to apples comparison, right? Also, another note is that the LTP targeted RNA-seq data is only considered qualitatively, which means that we only look to the presence or absence of fusion transcripts. We will not look at the expression levels. At least that's not the current intent of the essay. But there may be something more there. And also, for the OGM side, we use so-called rare variant algorithm, not the genomic assembly, particularly for CRLF2 type of aberrations, which may handicap OGM, but we haven't validated the genomic assembly in our laboratory, so yet we are working on it. And that's the reason that we use rare variant algorithm. And now, before wrapping up, let me remind the conclusions. Well, there's a high rate of concordance with OGM LTP, which is reassuring. The genes with enhancer hijacking has much lower detection rate with LTP. Particularly the MICOM is a class-defining abnormality with a very bad prognosis for AML, or IGH rearrangements in BLL, and the BCL11B rearrangements, which is a driver for TLL. We should consider alternative methods to RNA-seq, such as OGM. Or, I mean, it can be, or if you are in a resource-poor environment, just fish for MICOM and IGH. OGM is certainly a reliable technique for detection of insertions. Also, interchromosome recombination can also lead to fusion transcripts. I'm show you the ones which are pathogenic, more pathogenic and more driver. But probably the aberration that always said that, okay, this is a read through transcript, so on and so forth, not all, but some, maybe due to interchromosome recombination, such as small deletions. And I should stress that there is no silver bullet. I mean, all techniques have its own limitations. And for laboratorians, well, we still have a job. I mean, it's not at least February 2026. We are needed in human beings, and I hope it will last at least until my retirement. Um, so this is the end of my presentation. I'm ready to take questions. Thank you.